Hi, my name is Olivia Walton,
And I'm Sara Mooney.
and we're on Appledore Island off the coast of Maine. And we're taking an intro biology course with a marine focus. This week, we're learning about a new subject called Evo Devo.
The new concept of Evo Devo has emerged from embryology and evolution. Evo Devo's overlying theory is that a species can adapt over a long period of time, like evolution, but also an individual can adapt during its development. The individual, or in some cases a community, can express certain genes that better adapt them to specific environmental stressors. Turning these genes on and off, or how much they are expressed, can result in completely different forms or qualities of an individual or subspecies. These genes are called toolkit genes. They can create a large phenotypic change without changing the genotype. The specific study of these toolkit genes is called epigenetics, which hopes to discover what different expressions of DNA have different functions. Olivia is going to talk to you more about examples of epigenetics, Evo Devo, and their implications.
There are many examples of Evo Devo all throughout the animal kingdom. One specific example has been around long before Evo Devo was even thought of. This is the well-known example of the evolution of Darwin's finches on the Galapagos Island. These finches look nothing much like the warbler finch found on the mainland of Ecuador. One finch, the ground finch, has evolved to have a very large nut-cracking beak, while the cactus finch has formed an elongated beak for drilling into fruits to get at the seeds. They both use genes that were already present in their bodies when pressed to find new ways to get food. The ground finch began to more strongly express the BMP4 gene in early development, which produces a protein that signals bone development, while the cactus finch expressed more calmodulin, which causes the beak to grow longer. This demonstrates an important concept of Evo Devo, which is that new forms of an organism can come from new uses of the toolkit genes, like BMP4 and calmodulin. Another very important concept of Evo Devo is that unrelated species can use the same gene to find similar solutions to the same ecological challenge. This can be seen in two separate lakes in Africa with very similar environments, Lake Malawi and Lake Tanganyika. In both lakes, there are two unrelated species of cichlid fishes that are found with long and flat-headed and a deep underbite. Both species have used the BMP4 gene to adapt in similar ways to their similar surroundings. There are also examples of Evo Devo in the human world. One example is the discovery of HIV-resistant sex workers in Nairobi. 140 workers out of the 2,000 were found to have higher levels of protein with antiviral and anti-inflammatory actions in their vaginal fluid. This seemed to give the women a resistance to the HIV virus. It was later found that if these women had reduced workloads, they became susceptible to catching the virus. Another group of HIV-resistant people were discovered in Scandinavia. Sara will now go into more detail about the resistant gene.
This gene that Olivia is talking about relates to a T cell within certain chemokine receptors called a CD4 T cell. Chemokines and chemokine receptors form a regulatory network controlling the development, recruitment, and activation of lymphocytes. CD4 T cells have CCR5 chemokine receptors. HIV gains entry into the immune system by binding to these specific CCR5 receptors and infiltrating the T cells. Some people, specifically those in Northern Europe, have a deletion or lack of expression of the gene that codes for the CCR5 receptor. This means the HIV virus can no longer exploit the cells in the body. The allele CCR5 delta 32 of the chemokine does this by introducing a premature stop codon, thus masking the receptor. Homozygous carriers of this gene are resistant, while heterozygous carriers have reduced viral loads and slower progression of AIDS. Using concepts of Evo Devo, scientists believe that this mutation is the result of a deletion in a single individual about 700 to 2,000 years ago in Viking populations before their dispersal around Europe. There are studies going on now trying to find out the cause of the deletion. Although the cause of the mutation is unknown, it is believed that the individual's genes were adapting to a stressor. Could this small tweak be changed in everyone? Are there many genes like this that could be used to prevent other diseases and viral infections? How can we use this new concept of Evo Devo to find the answers to these problems?
In our opinion, Evo Devo is a very new field with unsurmountable potential. Many studies on diseases that plague our world have already begun and will continue, bringing us new solutions and answers to previously unanswered questions. Only the future will truly show the possibilities that come with Evo Devo. Thank you.